Hello, everyone. Just wait another minute while everyone's joining. A few people in the waiting room. There we are. Oh, it's great to see so many people here. Thank you, everyone, for taking the time this evening to come to the talk. It's going to be a really interesting one. For anyone who doesn't know me, I'm Beth, the support line nurse for WMUK, and we're joined tonight by Dr. Gina Doody, who is an associate professor at Leeds University. She's talking to us about research in B cells and how it relates to WM, and she's really kindly giving up her time to do that. Just a little bit of housekeeping before we start. We'll be recording this so we can put it up on the website afterwards, so people who couldn't make it can still watch it. I'd also ask everyone if you can check they're on mute while they're not talking. So there should be a red line through your microphone icon, just to make sure you're on mute while someone else is talking. And also there'll be time for questions at the end, but Dr. Doody says she's happy because it's quite a complicated but really interesting subject. She's happy if you want to ask questions in the middle while she's talking, then just raise your hand virtually by pressing the raise hand icon, or you can write in the chat that you want to ask a question and I can call you in. But as I say, there'll be plenty of time at the end for people to ask questions. So without further ado, I'll hand over to Dr. Doody.
That's great. Thank you very much, Beth, and good evening to everyone out there. Thank you very much for coming to listen to me talk about B cells, which are my favorite cell in the body. So I'm currently based at the University of Leeds, but as you can probably gather from my accent, I'm not a native of Yorkshire. So I'm actually from the state of Maryland in the US. And I started my PhD back in the States quite a number of years ago now, so more than 30 years. And I was doing my degree in Baltimore at Johns Hopkins School of Medicine. And it so happened that my supervisor decided to move to Cambridge. And I thought, why not? So I went with him. There were a few of us that moved to the UK. And so I transitioned to doing my degree in Cambridge for a number of years. And all that time, I was studying fundamental principles about how B cells behave. And then I stayed on in Cambridge for a little while longer at a place called the Baberham Institute, which is a research institute just outside of the city of Cambridge. And there I worked for a few more years studying B cells in a different laboratory. At that time, I actually met my husband in the lab. So we were both doing our PhD in the same lab and ended up getting married. And then we moved up north and are based at the St. James's campus at the University of Leeds. So if any of you are familiar with Leeds, there are two big hospitals in the city. So one is is in the center of the town where all the shopping is, and nice location. And that's also where the university is based. But we're on the other side of town over at St. James's. So we work in this building, this tall building here called the Wellcome Trust Brenner Building. And those of you following the news, the riots happened just over here, a few streets away. So Ruben and I, my husband, have been basically running the lab together for over 20 years. So I'm not a clinician. My background is in pure research, whereas Ruben spends 50% of his time doing clinical work. So he's a histopathologist by training and spends all of his time diagnosing blood cancers. So that's a little bit about my background. And so now the rest of the time, I'm going to be talking to you about the wonderful cells that are B cells and the importance to them in your immune system. And then what happens when they go a bit rogue and contribute to tumorigenesis? So just starting at a very basic level, your immune system can be divided into sort of three different levels that are providing you with protection. So the first level is provided by barriers such as your skin or the hairs in your nose, which keep most invaders from entering into your body. The second level is something that we're all born with, and this is called innate immunity. This provides an immediate response to some kind of foreign invader. An example would be a neutrophil, a cell type called a neutrophil, which hunts and kills bacteria. The third level is known as adaptive or acquired immune response. And as the name implies, this is a type of immunity that can change over time. It adapts. So it's acquired after exposure to a foreign invader. So while the innate immune response is immediate, the adaptive immune response takes time. There's a delay, but it's also highly specific to the foreign invaders that you're infected with. This is why every year you have to get a flu shot, because the flu virus is constantly changing, and our immune system is so specific that unless we get a new vaccination, our bodies won't be able to recognize it. Innate immunity is comprised of lots of different cell types, and I mentioned the neutrophil before, which can hunt down and kill bacteria. On the other side is adaptive immunity, and there's two separate branches of this. One type is carried out by cells, and in particular involves something called a T cell. The T stands for thymus because these cells develop in the thymus. The other branch is known as humoral immunity, which is basically carried out by protein rather than the cells. These proteins are called antibodies or immunoglobulins. It's called humoral because the antibodies can be secreted into fluids or humors of our body, which is basically blood. The antibodies come from another cell type, which is called a B cell. However, in order to make the antibodies, the B cell needs to change into something called a plasma cell. Plasma cells are dedicated to making very large amounts of antibody. So you can kind of think of them as an antibody factory. An antibody is the secreted form, whereas the term immunoglobulin can refer to either the protein on the surface of a B cell or the secreted form. But they are, broadly speaking, the same protein. And the antibody on the surface of the cell is able to sense specific structures in the environment. So the antibodies are very specific and recognize unique structures on invaders such as bacteria or viruses. The part that is recognized is called an antigen. So on this slide, you can see these little yellow dots on the protruding from the virus. And the very ends of the antibody molecules, these little tips, will specifically recognize that yellow area. So that's the antigen that it recognizes. Although plasma cells make large amounts of antibody, a single plasma cell will only produce a single kind of antibody. It only recognizes one thing. So in this picture, these blue antibodies, which recognize the bacteria, come from one plasma cell, which made lots of them, whereas these red antibodies, which recognize the virus, come from a different plasma cell. So each tip of those Y-shaped molecules binds to a particular part of the antigen. And since there's two of them, it allows them to bind together with precision. So using this mechanism, the antibodies can effectively tag a pathogen, like these viruses or bacteria. And this then allows an infected cell or a pathogen to be attacked by other parts of the immune system, or the antibodies can neutralize the invaders directly. To allow the immune system to recognize millions of different antigens that are found on all kinds of different pathogens, these little tip areas, the antigen recognition site of the antibody, come in an equally wide variety. The reason for this has to do with the genes that make up the immunoglobulin. In humans, there's a total of 176 different immunoglobulin genes, which can contribute and combine in a random fashion to make up the parts of the immunoglobulin molecule. So a good way of perhaps thinking about this is that it's like having a deck of cards. So you have 52 different cards, and they can give you a variety of hands. So if you're playing poker and you're dealt 5 cards, you can have a wide variety of different hands. Sometimes the hand is brilliant, sometimes it's just okay, and sometimes it's just plain old rubbish. It's the same with antibodies. So sometimes they have a very strong recognition of the invader. Sometimes it's only passable. And sometimes it's not actually good at all. The process of combining the genes occurs in a very particular sequence. So it's a very ordered process. This happens in newly made B cells that are found in the bone marrow. And you can kind of liken it to the early development of a child. So it goes through various key stages with milestones before it progresses to the next stage. And once the immunoglobulin is fully assembled, it can be found on the surface of the cell. At this stage, the B cells are called immature B cells, but they're ready to be educated and turn into a more mature B cell. As part of this journey to become more mature, the B cells leave the bone marrow. So as probably many of you know, the bone marrow is the central region of different bones. So sort of a spongy area that's protected by the hard bit that we traditionally think of as the bone. So once you've made these immature B cells, they can come out and travel to different lymphoid organs, such as lymph nodes that you can find in places such as your armpits or the groin area. They can also go to other lymphoid organs, such as tonsils or the spleen. Lymph nodes are small bean-shaped organs that are about the size of a pea normally. A lymph node's job is to filter substances in your lymph fluid. And this is a collection of fluid that drains from your cells and your tissues. Lymph fluid can contain things like proteins, minerals, fats, nutrients, different types of white blood cells, but it can also include damaged cells or cells that are dying, as well as foreign invaders. So the lymph fluid flows through the tissue that makes up the lymph nodes, and there the tissue filters and recycles the lymph fluid that your body needs. The cells within your lymph node, including B cells, will attack, destroy, and eliminate waste or sort of things like those dying cells that might come through, and especially foreign invaders to help your immune system keep you healthy. To enable B cells to carry out this job, they are covered in a wide range of proteins that sit on the cell's surface that help them decide what to do. So the kinds of things that a B cell might be considering doing is to decide, am I in the right place? Is what I'm seeing a pathogen, or is it something that's not so toxic? Should I be mounting a response? If I'm already undergoing a response, should I change the type of response I'm making? Do I need to make a copy of myself? Should I turn into a plasma cell to make antibody? Or should, in fact, should I go someplace else? Or maybe the cell's not being very productive and or it might be damaged and has to undergo cell death. So these are all the different kinds of questions that a B cell needs to address. To make these decisions, the B cell relies on interpreting signals that come from outside of the cell, using all of those surface proteins and lots of other helper proteins that relay the information into the control center or the nucleus of the cell. The first step is that the surface receptors or surface proteins recognize a specific signal from the environment. So you can also call this the primary messenger or a ligand. This can lead to the recruitment of an army of second messengers that relay the signal inwards. You might even think of it as something like a stack of Legos. So you get one kind of connecting to another all the way inside the cell. Then there are proteins in the nucleus or this control center that help interpret the message and make changes in the genes that the cells express. And in any given cell, only a fraction of the genes are ever turned on. And gene expression is the process by which these genes are activated to produce the required proteins. So this change in gene expression will lead to a change in behavior from the cell. So one set of signals might tell the cell to completely stop what it's doing. Another set might say slow down and wait. Or yet another set of signals tell the cell to proceed full steam ahead. For a B cell, the most important signal is the recognition of the antigen on a foreign invader. This is why the immunoglobulin molecule on the surface of the B cell is also referred to as the B cell antigen receptor, because it's a receptor that can see and recognize antigen. Although the immunoglobulin is the protein that identifies the foreign invader, it needs help from other surface proteins to start the relay of information into the cell. So in immunology, you'll see lots of proteins that are called CD and then a number. So I'm sure you may have seen it in your clinical reports. There's lots of them. There's over 300 different CD molecules. The letters CD stands for cluster of differentiation. This is an experimental technique that has been used to identify and to investigate different cell surface molecules on different cell types. These are then given numbers in the order that they were identified. So on this picture, I'm showing CD79, CD19, and CD22. So out of these, 19 was identified first, followed closely after by CD22, and then much later, scientists found CD79. Once the B cell receptor receives a signal, so it's recognized something like a virus, it can be interpreted in different ways to carry out different tasks, such as interacting with T cells or turning into plasma cells to produce antibodies. In order to achieve these outcomes, the B cell receptor uses a wide variety of second messengers to relay the information into the nucleus. And they all have different names and slightly different functions. One very important secondary messenger is a protein called BTK. So that's this one shown in yellow. And it sits at kind of almost like a top of a pyramid and controls multiple different pathways. So multiple different relays of information into the nucleus. And it's absolutely essential for B cell function. The importance of BTK can be seen in patients with a rare genetic disorder called X-linked agammaglobulinemia. These patients, so it's a rare disorder, and these patients are born without the BTK protein. They fail to make B cells or produce antibodies. And as a result, patients with untreated disease are prone to develop serious and even fatal infections. The antigen receptor alerts the B cell to the fact that there is a very specific foreign invader present. However, there are other proteins that can give a more general warning of danger. A group of proteins called toll-like receptors can detect parts of pathogens. So whether it's maybe some fungus or bacteria or viruses, there are very specific toll-like receptors that can tell the cells that there's danger nearby. For example, a B cell can become aware of the presence of viruses when these types of receptors come in contact with fragments of DNA from the virus. B cells have a wide variety of toll-like receptors on the cell surface, and they're very sensitive to encountering the signal that triggers these receptors. After such an encounter, this leads to enhancements in B cell survival and antibody production. The toll-like receptors also initiate a relay of information to make these changes in B cell behavior. An important protein that helps toll-like receptors is something called MYD88. And you'll see also BTK plays a part in bringing the signal into the cell. If a B cell in something like a lymph node encounters a foreign invader and transmits the correct signals, then a very dramatic change can occur. The B cell knows it's the right one for the job and makes lots and lots of copies of itself so that the response will be magnified. The B cell also makes changes in the immunoglobulin molecule to try to make it more efficient. This happens in a particular structure in the lymph node, which is called a germinal center. You can think of a germinal center as a kind of training center for B cells. At the start, they're considered naive B cells because they haven't encountered a pathogen before. Once they see the pathogen, then they go through a sort of boot camp to improve their fitness. The B cells that don't meet the standards are removed, but the ones that are improved following the germinal center training are selected for new roles. One of the things that happens during the germinal center is the intentional introduction of mutations into the immunoglobulin genes. The idea behind this is that some of the mutations will introduce changes in the protein structure in this tip region that comes in contact with the pathogen, so that it will allow binding or recognition of the pathogen with increased efficiency. So if we go back to the previous comparison to having been dealt maybe just an okay pair of hand of cards with maybe one pair, you could trade in some of your cards to see if you can improve your chance of being a winner. So it's a similar sort of thing with the immunoglobulin. It makes changes in the structure with the hope of having a better way of seeing the pathogens. In the germinal center, the immunoglobulin can also be altered in the tail part of the Y-shaped structure. There are several different variations, which are then called isotypes or classes. Antibodies of different isotypes are adapted to function in different compartments of the body. The first antibodies to be produced in an immune response are always IgM, which forms this pentamer structure. So it's got 5 different immunoglobulin or antibody molecules joined together in the center. And these are mainly found in the blood, and they're less efficient generally at recognizing pathogens. In contrast, other isotypes like IgG are much smaller, so they can move more easily from the blood into tissues. So IgG is the main isotype found in blood and the fluid that surrounds cells and tissues. And generally, it has a much better efficiency of recognizing pathogens. And so it's produced later in an immune response. Then you have these other types as well. So something like IgA is the principal isotype in secretions like mucus or saliva or tears. And so it can protect against pathogens in your digestive tract. And then IgE is another form, and this has specific types of responses against parasites or worms. If a B cell successfully completes the germinal center and makes an immunoglobulin with increased usefulness, it's now a more mature B cell. So it's grown up and is more mature and can take its place in the immune system, ready to join the workforce. So with the 2 main types of jobs that you get after you come out of the germinal center, a B cell can turn into what's called a memory B cell. And this remembers the antigen that's been seen before. So your immune system can quickly identify and fight it in the future. On the other hand, a B cell can turn into a plasma cell, and it pumps out large quantities of antibody. When these new cell types come out of the germinal center, they travel to different parts of the body to carry out their new roles. So the memory B cells recirculate into other lymphoid organs on high alert for specific pathogens, while plasma cells generally travel to the bone marrow. This requires another set of signals that the cells have to respond to. The plasma cells need to travel from the lymph node to the bone marrow. Then they need to find the right sorts of cells, so sort of a neighboring support group, that provides signals to keep them alive. This is particularly important for plasma cells as they can actually last for the lifetime of an individual. For example, you may be amongst the generation who received a smallpox vaccine when you were a child. And although you've never been exposed to smallpox, you're likely to still have antibodies in your blood that can recognize the smallpox virus. These are produced by plasma cells that were made when you were vaccinated as a youngster and have survived all that time in the bone marrow. One of the most important signals that guides a plasma cell to the bone marrow is generated through a protein called CXCR4 on the cell surface. it senses a very small protein that acts as an attractant to lure the plasma cells into the bone marrow, much like a bee is attracted to a flower. Not only does CXCR4 guide the cells to their destination, but it's also important for keeping them there. Once CXCR4 receives its signal, it relays this information into the cell to change behavior. But also, the CXCR4 gets pulled inside the cell. This is to let the cell know that the signal has been received. Once it's inside the cell, it can have two different types of fates. So it can be destroyed. So that signals the cell that it needs to stop. It's had enough signal. Or it can be recycled back onto the surface, and then it can undergo another round of receiving signals and relaying that information back into the cell to change behavior. And once again, this protein BTK plays a role in helping the CXCR4 signal to occur. So if you consider all the ways that BTK contributes to relaying different types of signals, it's clear just why it's so critical to B cells throughout their entire life cycle. From the time that they're developing B cells and they're making their B cell receptor, once they become more mature and need to respond to pathogens, and then also once they've become plasma cells and need to go to the right place and receive the right signals for survival. So those are some of the fundamental features of what happens to a B cell during a normal immune response. So where does it all go wrong and end up making a B cell cancer? One of the major hazards that a B cell faces is the germinal center. In many ways, the B cells that are in the germinal center already behave as if they were cancer cells. For instance, B cells undergo massive proliferation and expansion of individual clones. Secondly, the changes to the immunoglobulin gene involve making breaks in the DNA, which can cause instability in the immunoglobulin genes as well as other genes. Normally, if dangerous DNA breaks are made, the cell stops and might actually die if the damage is too severe. But germinal center cells largely ignore the problem. Finally, the germinal center B cells consume energy in a different way to help with their rapid growth, which is very similar to how a cancer cell behaves. Because what the B cells are doing in the germinal center is a dangerous business, there's a higher chance that unwanted DNA mutations can occur, and that these hazardous changes will be tolerated. The unwanted mutations often occur in genes that are critically involved in the regulation of all those things the B cell needs to be doing. So proliferation, death, turning into a plasma cell. So these errors can result during the antibody gene modifications. Therefore, the production of an improved antibody comes at the risk of converting into a cancer cell. And that's why approximately 90% of all immune system cancers arise during the process of a B cell becoming a plasma cell. Different types of B cell cancers shown across the top here. So, for example, Burkitt lymphoma, chronic lymphocytic leukemia, follicular lymphoma, and then diffuse large B-cell lymphoma have characteristic genetic lesions, so DNA mutations, that make them behave differently from one another. The type of genetic lesion is usually related to what the cell was doing at the time the error occurred. And this can occur at multiple stages during the germinal center, such that different types of B cell cancers originate from cells undergoing a particular part of this training regimen. The presence of the genetic lesions essentially freezes the cancerous B cells so that they closely resemble their original cell type. And importantly, most of the time, they cannot move forward to become a plasma cell, so they're stuck in the same state. The presence of the genetic lesions also makes the mutant B cells more likely to generate additional copies of itself, and they're better at surviving. The tumors then result from the multiplication of genetically identical cell clones. Waldenstrom cells are thought to come from a memory B cell that is finished going through the germinal center and has been exposed to that dangerous process. However, unlike other B cell cancers, Waldenstrom B cells can still go on to make plasma cells that usually secrete IgM antibodies. Another unusual feature of Waldenstrom B cells is that they accumulate in the bone marrow rather than traveling through a lymph node. So while most B cell cancers resemble a single point in the B cell life cycle, Waldenstrom's is a disease with multiple cell types. And these cell types have different characteristics and behaviors. For example, B cells have a protein called CD20 on the surface, but this is usually no longer present on a plasma cell. Another major difference is that B cells are normally capable of reproducing themselves, but this function is normally lost on plasma cells. And then yet another distinction is that the B cells don't actually produce any of the IgM antibodies, and this is restricted to cells of the plasma cell type. The different cell types in Waldenstrom's also causes different problems. The Waldenstrom B cells can fill up the bone marrow, crowding out the space and disturbing the normal process of making new blood cells, which also occurs in this area. On the other hand, the Waldenstrom plasma cells secrete large amounts of IgM antibody that can lead to numerous clinical problems, such as hyperviscosity syndrome or tissue deposition of IgM, or the autoantibody activity of this molecule. A unique feature of Waldenstrom's is that almost all of the cases, so greater than 90%, are caused by a mutation in the gene MYD88, which changes, in almost all cases, changes an amino acid called leucine, which is this L, to a different amino acid called proline, which is this P. And it occurs at the same spot in the protein. So it's this position called 265. So that's the number of amino acids in the protein. As mentioned previously, the MYD88 protein acts as a messenger to relay information from toll-like receptors. However, if the mutation is present, the signal relay happens without needing to be exposed to a pathogen danger signal. The cell then behaves as though it's getting ready to mount a response and increases its capacity for proliferation and survival. In addition, an additional 30% of Waldenstrom patients, the CXCR4 gene is also mutated. This can generally results in cutting off this little tail on the end of the CXCR4 protein. And this means that it can no longer be pulled into the cell after receiving a signal. So it just keeps going on and on. This also tells the cell to keep growing, and it helps it stay in the bone marrow. There are some additional more rare mutations that have been found in Waldenstrom patients. And it's likely that the presence of these additional genetic lesions can alter the behavior of the cancer cells as well as the response to treatment. Up until recently, the standard treatment for Waldenstrom's involved traditional chemotherapy agents such as bendamustine. But now there's a much better range of treatment options that include monoclonal antibody therapies such as rituximab and novel agents that interfere with different aspects of B cell function. And the most effective class to date are the BTK inhibitors, ibrutinib and now zanubrutinib. The selection of an optimal therapeutic regimen is an individualized decision that's reached by the clinician and the patient. It has to consider a variety of factors, including patient characteristics, so whether you have other medical problems or taking other medications, the disease characteristics, so for example, the mutational status, or looking at prognostic markers, and also the effects of the treatment themselves, so some are more toxic than others. As an example, in the presence of the CXCR4 mutation, this reduces the effectiveness of treatment with ibrutinib. Although this is not as diminished in the case of zanubrutinib, so that seems to be an improvement over this lack of response with ibrutinib. Another aspect to consider is how the treatment affects the different cell populations in Waldenstrom's. And I'll give you an example of what I mean. So the monoclonal antibody therapy, rituximab, targets the surface protein CD20, which I mentioned just a moment ago, is found only on the surface of B cells. So this, and as they become plasma cells, it's usually no longer found on the cell surface. So while rituximab is very effective at eliminating the B cell component of the disease, it spares the plasma cells, which are the producers of IgM that give you all those clinical symptoms. But this is also why BTK inhibitors are so successful. Since BTK is used throughout the B cell life cycle, blocking its function can potentially eliminate all of the cell types that are present in Waldenstrom's. So that gives you a flavor of the nature of B cells and how they relate to Waldenstrom macroglobulinemia. So how do we use this information in the laboratory to learn more about the disease and help improve the effectiveness of treatments? Our lab has devoted a number of years to working out a model that tries to mimic the various stages that a B cell goes through on its journey to becoming a plasma cell. So in the lab, we start with blood samples from healthy donors, usually from people like me, so I donate all the time to our team that works in the lab. Or we take them from people with Waldenstrom's, and in that case, we're taking bone marrow samples. And we can isolate the B cells from blood or bone marrow. And then these B cells are exposed to signals that trigger the B cell receptor, the toll-like receptors, and as well as other receptors that tell the B cell that it needs to start changing. Over time, we provide the B cell with different types of signals to push it into becoming a plasma cell. And once it reaches the plasma cell stage, we give it the signals it needs to survive. Throughout the experiment, we keep track of what's happening to the cells. For example, we look at the proteins that are on the surface of the cell. And we do this using a technique called flow cytometry. And I apologize, I haven't put in a slide that explains that, but many of you may be somewhat familiar with this in that it's used for clinical diagnostic purposes to look at what types of cells are present in your blood or bone marrow. So we can use this to monitor the types of patterns of surface proteins that the cells have. So when we start our experiments, a B cell will have a high level of CD20 and also this protein CD19. But over time, it loses the expression of CD20. So we see it going from this corner up here, the upper right of this way of mapping the surface proteins. And over time, it moves out of that corner and moves across. So that means it no longer has CD20 on the surface. On the other hand, as the cells become plasma cells, they start to express some new proteins on the surface. So the ones shown over here, so CD38 and 138. So in a kind of intermediate phase, it starts to express CD38. And then when it's a fully mature plasma cell, it expresses the protein 138. So we can track what's happening to the cells based on the proteins that they have on their surface. At the same time, we can also check what's happening to the various genes that are expressed. So B cells have a characteristic pattern of genes that they have on, but this changes over time. And when we compare the types of genes that are expressed in the cells that we make in the lab, they look very much like the genes that you would find in a plasma cell that's taken straight out of the bone marrow. And also, if you look at the cells under a microscope, they look like a plasma cell, so they've got a very characteristic shape that tells us that they look as a plasma cell should. So using this system, we can reproducibly make plasma cells from healthy donors over and over again. So we've had lots of people working in the lab on projects like this for many years. And this slide shows experiments done with B cells from 12 different people. I'm sure one of them was me. And we can track the surface proteins over time. And essentially what we can see is that they all look remarkably similar. So you can see CD20 behaves the same way, CD38 the same way, and CD138. So this is very good at making normal plasma cells. If we do the same type of experiment using Waldenstrom B cells, plasma cells can be made, but the pattern of surface proteins is not uniform in all of the samples. So previously, all of the healthy ones were all quite closely clustered, so they all look very similar. But in this group of 7 Waldenstrom patients, you can see that there's quite a range of these proteins, so they don't all look exactly the same. Oops. Oh, I think I've just been signed out of my, hold on. Shall I keep going? And this slide shows some of the differences that we observed. For example, although the plasma cells made from healthy donors have very little CD20 on the surface, the Waldenstrom cells still have some CD20. And down at the bottom, it shows that although the healthy cells make CD38, some of the Waldenstrom cells aren't very good at making this protein. So we can observe that there are different patterns of surface proteins from the different patient samples. How can we use this information? Does it relate to the genetic lesions that are present in the cells? Can we use it to predict response to treatment? For example, maybe some patients will respond better to rituximab if they have CD20 on their plasma cells. To tackle the first question, we're doing some different types of experiments that involve purposely introducing the genetic lesions into healthy B cells to try to turn them into cells that look like Waldenstrom's. We have a student who's been working on this, and he successfully introduced the mutant MYD88 protein into healthy B cells. When he does this, they tend to have more CD20 on the surface than control healthy cells. Also, if we grow the cells in conditions that lack the survival factors, the healthy cells die, but the cells with the mutant MYD88 can still survive. So we think we have made cells that are starting to resemble real Waldenstrom cells. So going forward, we'd like to test how different combinations of the genetic lesions, such as the CXCR4 mutation, alter the pattern of surface proteins and the ability of the cells to grow and survive. We'll also test how these different combinations affect how the cells respond to therapies. So in conclusion, signaling controls cell maintenance, activation, and survival. And the signaling molecules are therapeutic targets that can be used to eliminate the tumor cells. BTK are particularly useful as a target since it's used in multiple pathways and differentiation stages. And in the lab, we can use model systems that can provide a useful means to study how individual cases of Waldenstrom's differ. So just as a final slide, I'd like to say a special thanks to the two students who worked on Waldenstrom's in the lab. So Jenny finished a number of years ago, and she was her project was actually partly funded by WMUK. So thank you very much. And Adam's a more recent student who's still working in the lab, and he's funded by the Ella Dickinson Trust. Also, some of you may know Dr. Roger Owen, who is a WM clinician, and Ruth DeToot, who runs the clinical samples here in Leeds, and just other funding bodies who've helped us along the way. Right, so that is the end of my presentation, and I'm happy to try to answer any questions that you may have.
That was fantastic. Thank you, Dr. Doody. A really difficult topic to condense down into a talk, but yeah, that was fantastic. So if anyone's got any questions, you can either write in the chat as someone has done now, I'll read those out, or you can press the raise hand icon and I'll call you in. So I'll just read out the one from the chat first. So are memory B cells destroyed during chemo, and or does your immune system reset?
Oh yeah, so it depends on what kind of chemotherapy. So memory B cells are generally not dividing. So they're going around looking for pathogens. So until they encounter the pathogen, they don't divide. So some types of chemotherapy don't really act very well on a memory B cell because it needs that signal first to make it divide. So typically, chemotherapy agents are best used when cells are very rapidly undergoing that process. So that's a normal memory B cell, but because Waldenstrom's isn't normal and it is undergoing those cell divisions, that's why chemotherapy is more appropriate for targeting that type of memory B cell. In terms of resetting your immune system, so yes, a lot of the treatments that will be given for treating any type of B cell cancer can affect your normal B cells as well. So during the process of having the therapy, this dampens your immune system and it does suppress some of your normal B cells, but these can reset. But I think there certainly are problems with this. And one thing that I think is going to be an emerging problem in the coming years is that people on therapies like zanubrutinib or rituximab may develop secondary immunodeficiencies, so that their immune system doesn't work as well as it should. And so that is a potential problem when you're having those treatments.
Okay, great. Gordon, do you want to ask your question?
Yes, please. Can you hear me okay?
Yes,
Yeah,
yes.
yeah.
Oh, really good. First time I've tried this headset out. Early on in your presentation, you mentioned the B cells and an involvement with the lymph nodes. I'm wondering if it also can affect the movement of lymphatic fluids. If it's maybe as it builds up in the bloodstream, it stops the lymphatic fluid from being absorbed back into the bloodstream through the normal process, or whether it stops it from being treated by the lymph nodes and maybe cause lymphedema.
Yeah, so you may, like having the high levels of IgM in your lymph. So that can impede the filtering. So, and having the IgM is a very bulky molecule. It's much bigger than the other classes of antibody, and that's why it can also get stuck in different tissues. So as it's traveling through, just because of its size, it can clog up things. So the kidney is particularly prone to having immunoglobulin get trapped in that organ.
So it could be causing lymphedema.
Yes, yeah.
Thank you. Thank you.
Okay, Joy, would you like to ask your question?
Yes, thank you. Thank you very much for that talk. It was brilliant. And I know which mutations and which genes I have. So now I know why, what they're doing. So thank you very much. That's been very clear. And I'll probably watch it again, the presentation again. My question was to do with diagnosis, because I speak to quite a few WMers who hate having bone marrow biopsies. I mean, I find them very unpleasant, but I don't hate them as much as a lot of people have. And it's certainly stopped people from entering clinical research trials that I've heard about. Is there going to be a future where we could have a biopsy, say, of germinal cells from the lymph nodes or something like that we could have instead?
Well, I suppose one thing to bear in mind is that Waldenstrom's is preceded by MGUS. So often you might have the early stages that perhaps we might be able to detect in blood if we, and certainly people are monitored that way. So you can, well, the immunoglobulin is quite a good predictor of what's happening. But whether we can detect, because the mutation is rare, and if you sample blood or lymph node, the chances of finding that cell there may be very low. So you may not actually be able to detect the mutation accurately. So it's a combination of whether the tests that we use are sensitive enough to find those very rare cells. And because the Waldenstrom cells don't really do very much in a lymph node, if you see it in a lymph node or in the blood, you don't know whether it's actually going to go on to cause full Waldenstrom's. So, for example, a group of patients who have Schnitzler's syndrome, which is can be a precursor state to Waldenstrom's, they can have the MYD88 mutation present, but they may never develop Waldenstrom's. So it's quite tricky. So you might have the mutation, but that doesn't always mean that you'll go on to develop a disease that needs treatment. And so it may come more to, are there better ways of accessing the bone marrow? So as surgical techniques improve, I think perhaps that might also be a way forward, that they're maybe less invasive, or better ways that are less painful. Because I know certainly, yes, it's not a pleasant procedure for anyone. And if you could do it via a blood sample, that would be certainly much preferable.
Thank you. It's a few years offward then.
I think so, yes.
Thank you.
Andrea in the chat asks, is it common for WMB cells to transform to other lymphomas like diffuse large B cell?
So it can happen, but it's thankfully very rare. So on the slide that I had with the more rare mutations in addition to the CXCR4, some of those, if those are expressed in the Waldenstrom clone, then yes, that can lead to progression to an more aggressive diffuse large B-cell lymphoma. Yes.
Sheena also asks, once BTK inhibitor like ibrutinib has failed, or rather WM has got past it, how far along are the new drugs in development for next stage of treatment?
Well, I think so. Luckily, there are lots of good drugs, and I think a lot of this has been pioneered in CLL, where they're doing lots of different combination therapies. So things like ibrutinib and venetoclax together, so the BCL2 inhibitor, which does have some nasty side effects, but I think they're getting to grips with the best way of administering those combinations. So yes, I think that they are certainly in the works, and they're coming through. So if ibrutinib is not successful, then there are other combinations that could be tried.
So I don't want to go too far past 8 o'clock, so I'll take questions from Keith, then Philip, and then Lucy, and then I'll wrap up for the evening. So Keith, do you want to ask your question?
Yes, thanks, Gina. Wonderful presentation. Some people remain on active monitoring for years without needing treatment. Have you done any checks or evaluations of those people to see why they can stop on active monitoring for that amount of time without requiring treatment?
No, so this is a puzzle, and I think it's likely to reflect the frequency that the B cells turn into those plasma cells and make the antibody. Because one of the things that's part of the unusual feature of Waldenstrom's is because it's in the bone marrow, it's not being exposed to the normal types of signals that trigger making plasma cells. So it's in fact probably less rapid or less efficient than would happen in a lymph node situation. So I think it has to do with the types of cells and the types of signals that the Waldenstrom B cell is exposed to. Because your bone marrow is changing all the time, it mostly is changing to provide all the different other types of blood cells that you need. So some of the things that are short-lived, like red blood cells or neutrophils, which you need to replace quite often. So the bone marrow changes the types of cells that are present every day. And so depending on what's present in the bone marrow, it might trigger Waldenstrom's to go down the plasma cell route, or they just do it very slowly. So it's difficult to know what exactly distinguishes one patient who doesn't progress and just has very sort of manageable levels of IgM to the ones who have a more aggressive type of disease.
Okay.
Thank you.
Want to ask a question?
Yes, thank you. Can you hear me all right?
Yes, yes.
Thank you very much for your talk. Unfortunately, I missed the beginning bit, but I'll listen fully back on the recording. I just have a question about paraprotein, and I just wondered whether there were any current theories about what I gather is a somewhat rare situation of where paraprotein seems to drop quite significantly for no apparent reason, i.e. with somebody who hasn't had any treatment. I remember some years ago going to a meeting where someone said that their I can't remember exactly what the figures were, but it was something like their paraprotein had dropped from about 20 to 10, i.e. very significant. And I just wondered if there were any current theories as to why that sort of thing might be happening. In that particular case, someone said, well, what's been happening to you recently? And the lady said, well, I'd had a knee operation. And she said, well, it might be something to do with the sort of different stresses that your body was under as a result of that operation. But I just wondered if there were if there's been any more light on that topic since.
So one thing that comes to mind, and this also relates to some of the emerging types of treatments. So some of you may have heard of CAR T cells, which are used certainly in other B-cell malignancies.
Yeah.
And the whole idea is using your or the immune system to attack the cancer cells. And in other types of cancers, so in more the solid tumors in other tissues like liver or some of the other ones, you can try to trick or try to boost the immune system so it can get rid of the cells without the need for chemotherapy or some of the other types of treatments. So it may well be that your immune system is capable of recognizing that the Waldenstrom cell should be got rid of. And so those T cells and the natural killer cells might be able to control and get rid of some of those cancerous cells. So there's a whole wide range of research done on what prevents or what kind of keeps those T cells or the NK cells from being able to do that job. And there are therapies to help the T cells try to mount that attack. So that's what I would suggest as one possibility for that.
Okay, thank you.
Okay, and Lucy, one last question from Lucy, and then we'll wrap up for the evening.
Thank you. I am still recovering. I'm just totally gobsmacked by, anyway, just a brilliant talk, and your slides were fantastic. I'm going to need to go over it several times to get the hang of what's going on. But to think there's all that going on in our bodies is just to be made aware of the intricacy. It's blown my mind, and thank you. When I was diagnosed from a bone marrow biopsy, well, not diagnosed, no one could actually tell me, yes, you've got Waldenstrom's. All they could say was, you might have Waldenstrom's, or it might be marginal zone lymphoma. So, and my diagnosis has been really made on a kind of probability thing because I've got MYD88 and because this. And so when you kind of analyze the cells, can you not kind of point at a cell and go, yes, that's a Waldenstrom cell? How do you recognize a Waldenstrom cell? That is my question.
Yeah, so that is tricky. And some of the cells have more characteristic features than others. So those kind of plots that I was showing you that have the surface proteins on them. So you can run a bone marrow sample through that kind of machine. And Waldenstrom cells do look different from healthy B cells. So you can kind of, so that's one of the ways that you then you use that bit of information along with the mutation analysis and the other kinds of things. So the split, so you're right. It's sometimes it can be hard to tell the two diseases apart. And actually, one of the things so just in the lab, we've also looked at splenic marginal zone B cells, and they can't make plasma. So they don't make plasma cells very well, unlike the Waldenstrom cells. So they've got some kind of block. They probably have other mutation. Well, they do have other mutations that are more like other B cell cancers, that they kind of get stuck and they can't make those plasma cells. So they don't. They're not churning out lots and lots of antibody like what happens in Waldenstrom's.
Yeah, I think I understand. thank you. I'll go over it all again. I'm really grateful. Thank you.
That's great, thank you. Yeah, it'll be going up on the website so we can all watch it again and go through it in more detail. But yeah, thank you again so much, Dr. Doody. There's loads of people in the chat thanking you and saying how informative and interesting it was. So thank you for giving up your time. We really appreciate it.
Well, it's been an absolute pleasure. Thank you.
Thank you everyone else for coming as well. As always, I'm on the support line if you've got any questions, but otherwise we'll advertise the next webinar soon and we'll talk to you soon.
All right. Good night.
Thanks, Dr. Doody. Thank you.
Thank you.
Thank you.
Very much indeed.